In the previous classes, we had studied the response of the host to various intracellular infections, the primary one being mycobacterium and the effects on fungus. In today's class, we will be studying two main types. One is viral infections and the other is new emerging diseases. And I think it is a fitting end to this part because you can see how the different host factors play an important role in modulating disease. This is a very important aspect, and you can see that there is no single mechanism. You have multiple mechanisms that are in place, and this is perhaps the reason why the immune system mechanisms are so redundant. You have innate, you have adaptive, and within each of these you have different subcompartments, right. Okay. So, if we go to the first part on viruses, so what are viruses? So, these are sort of inert particles, proteinaceous particles which replicate based on plant cells or animal cells. In our case, it would be mainly animal cells. And what are some of the things they do is that they use the nucleic acid and protein biosynthetic machineries to replicate in the host. Life is about replication because you would need to increase your progeny so that you can spread it. That is what biology is all about, and if you are unable to do it, then it does not quite work out. So, it is very important for pathogens or organisms to find mechanisms by which they can replicate. It is also important for the host to take care of its self and prevent this takeover by different pathogens, including viruses, and this interaction between the two is what we study in this part known as the immune system. One of the ways by which viruses infect is by utilizing cell surface molecules to gain entry in. So, essentially if we were to take a look at the different steps in terms of viruses, there are mainly four. One is attachment. A virus attaches to the surface of a specific cell. Often it is through a particular receptor, binding of a particular receptor. The most famous of course is your HIV virus, which you are I am sure you have heard of, where GP120 of the virus attaches to CD4 and that sort of helps in the entry of it. Now, there are co-receptors involved in this. We will be studying some of those chemokine receptors that are involved in this, but the specific interaction is between a viral protein and a specific receptor on the host cell. Now, after attaching an entry, you would the DNA or RNA, whatever type the virus belongs to, is injected into the cell. You need to copy, and as I have said, copying is very important because replication is the hallmark of life. Viral nucleic acids take control and begin to make new virus particles. So, what happens is if it is RNA, then it is converted into DNA and then back into RNA and ultimately into proteins, and if it is DNA, then back into RNA and then into proteins. Now, the viral particles are formed and then the cell bursts open and releases hundreds of these, and with the hope that some of these will be able to find host cells by which in which they can replicate and spread the progeny. Okay. Now, in terms of evading the immune system, you have different viruses that have got different mechanisms or have used different mechanisms. For example, the hepatitis C virus inhibits interferons. We will be discussing a little bit about interferons by inhibiting the action of PKR or protein kinase R. HSV or herpes simplex viruses decreases expression of MHC class I, avoids CTL elimination. Cytomegaloviruses also decreases expression of MHC class I. So, what has happened is that since the MHC class I response is so important in terms of infecting in terms of fighting viruses, the virus particles have evolved in such a way that means the species that are able to downregulate MHC class I are more have a more likelihood of spreading, and so that is what has happened is the evolution, the environment condition is such is that you are looking for progeny that can bypass the immune system, and one of the best ways by which they can do it is to try and decrease MHC class I. You will get a lowered CD8 response, and that helps. Then you have HIV, which decreases MHC class II expression. Then you don't have the large majority of your CD4 positive cells are destroyed, and as a result of which the helper activity is less, and so therefore the CTL the CTLs that are generated are often not fully functional. In case of influenza, it is a little bit different because they keep changing the antigens, and that is goes by antigenic drift or shift. Now, one of the important ways by which the host controls viruses is via the production of interferons. So, we will spend a few slides on interferons. Now, interferons were discovered several years ago in the 1950s by Isaac and Lindelman, because they found a factor that interfered, or a host factor that interfered with the replication of viruses. Subsequently, these were purified, and we now know there are three broad types. Interferon alpha is produced by leukocytes, interferon beta by fibroblasts. Both alpha and beta bind to a particular receptor known as the type 1 interferon receptor. Now, type 2 is interferon gamma, which is produced by T cells and natural killer cells. And the receptor for the interferon gamma is distinct from that of interferon alpha and beta. So, these two have different signal transduction pathways, and we will discuss that a little bit. You have natural killer cells, which will lyse a wide variety of virally infected cells, because if you remember, natural killer cells kill cells that where you have lowered MHC class I. Also, NK cells have NK receptors, and you have different types of whether they are inhibitory or activating type, and in case NK ligands are expressed on these virally infected cells, NK cells will kill these virally infected cells. Okay. So, a little bit about interferons. Now, interferons are transcriptionally regulated cytokines, and they play a very important role in modulating immune responses, especially antiviral immune responses. What happens in this case, based upon recognition, the toll-like receptors on dendritic cells and high amounts of interferons are expressed by dendritic cells. You have other types of cells also that were shown that can produce interferons. What this does is the production of interferons modulates the adaptive immune response, and that is the most important part. Now, as a result of this effect of interferons, viruses have developed different strategies to counter it. But in general, as I mentioned, there are two types of interferons, type 1, the type IFN alpha beta, and these are primarily antiviral in nature. They are involved in dendritic cell maturation, NK cell cytotoxicity, they increase NK cell cytotoxicity, neutrophil migration, and if you look at the phenotype of the receptor of individuals that lack the type 1 interferon receptors, they are highly susceptible to viral infections, and they have elevated levels of myeloid lineage cells. That is primarily because the interferons are probably inhibiting this particular lineage cells, and in the absence of it, these cells sort of take off. So, they might have some growth regulatory properties, but the primary effects of type 1 interferons are antiviral, and that is how they were discovered, and it is very important that we understand that. The other type of interferon, which is the interferon gamma, it is produced by activated T cells and NK cells, as we mentioned. They are immunomodulatory. They are very important for host defense. So, as was previously discussed, interferon gamma lacking mice are highly susceptible to infections by intracellular pathogens like Salmonella, Mycobacteria. We also talked about BCGiosis, if you remember correctly, in the past lecture. Now, interferon gamma knockout mice are highly sensitive to intracellular pathogens. Okay. A little bit about their signal transduction pathways. What is shown over here is the type 1 signal transduction pathway is shown here, and over here interferons function through what is known as the JAK-STAT pathway. JAK stands for Janus kinase, and is a tyrosine kinase, and STAT is a signal signal transducer and activator of transcription. So, what happens over here and what will be shown is upon binding about interferon with its receptor, you have activation of the JAK or the Janus kinase, and which phosphorylates the STATs. The STATs upon phosphorylation, they bind with each other or bind with other molecules, and they enter the nucleus where they transcribe several genes. So, the mechanisms by which interferons function is primarily through transcriptional regulation, and how this works is via through the JAK-STAT pathway. Very important. I will just briefly mention. So, here you have the type 1 interferon receptors. There are two subunits over here, IFN-AR1 and they are 2, and these are associated with the JAK1 and TYK2. Both these two are tyrosine kinases. Now, these two will bind to the STAT1 and STAT2 upon phosphorylation. The STAT1 STAT2 dimerize, they bind together with another molecule known as P48, enter the nucleus, and they form this complex known as ISGF3, and they will bind to ISRE or interferon responsive elements which is present in certain genes. For the ones that are shown over here are, this is the MX GTPases, MHC class 1, and the oligoadenolide synthase, which is important in antiviral responses. Okay. So, it is very important that interferons, you understand the way interferons function. It is mainly via transcriptional regulation, and that transcription of different genes that are involved in the response, and this functions through the JAK and the JAK-STAT pathway. The other interferon that we talked about, the type 2 interferon, was interferon gamma, and this binds to again the interferon gamma subunits, the IFN gamma R1, R2, and over here the tyrosine kinases, you have JAK1 and JAK2. Now, JAK1, JAK2 upon coming together after binding to interferon gamma, what happens over here, you have phosphorylation of STAT1, the STAT1 dimerizes and then enters the nucleus, and it enters the nucleus to form a gamma activated factor. So, the gamma activated factor binds to the gamma activated sequence, which is present in the promoters of several primary responsive genes like IRF1, P48, TAP1. And P48 is the same one that is present over here, and the interferon responsive factor goes and then binds to other genes which is shown, and they are a part of the secondary response. Okay. So, with respect to interferon gamma, you have two types of responses. Again, you have the JAK-STAT pathway plus playing an important role, but over here you have the primary response, and IRF1 then goes on and binds to other in the promoters. It's also another, it's a transcription factor. It is and it binds to other promoters of other genes, and which are part of the secondary responsive. So, you can differentiate the primary and the secondary responsive, because the secondary responsive genes will be sensitive, will require protein synthesis, and so therefore will be sensitive to cyclohexamide treatment. Whereas IRF1, P48, TAP1 for example, these are primary responsive and will be insensitive to cyclohexamide treatment. So, it is very important that students understand primary and secondary responsive genes and the way of differentiating them using a protein synthesis inhibitor. You could also use actinomycin D, which is RNA transcription inhibitor. Okay. So, we will now move to let me get this part. Yeah. Okay. It's not coming. Okay. Okay. So, now if we look at specific immune response to viruses, there are interferons is certainly one, but as mentioned, the immune system has different pathways that play an important role. And you have, it's a combination of both humoral and cell mediated. Now, in the humoral mediated one, a very important aspect is the generation of neutralizing antibodies. If you are able to generate neutralizing antibodies against the particular virus or bacteria, then you have a good chance that you will be able to mediate protective protective effects. And as was mentioned, that most of the effects, most of the good vaccines are the ones that generate very good neutralizing antibody. In this case, smallpox is a good example, and the fact that we have been able to eradicate smallpox is because of the fact that we generate that you are able, or one is able to generate a good neutralizing antibody against this particular virus. Okay. So the ability of good neutralizing antibodies is really key, so what these antibodies do is that they would bind to the viral capsid particles, they would inhibit the binding of them to its receptors, and more importantly it would allow they would fix complement, and as a result of which complement mediated lysis could take place. It would also help in opsonization, where antibody bound viral particles would be taken up by macrophages or phagocytic cells and phagocytosed. So there are different mechanisms by which this works. And some of the ones are shown over here. If the receptor is blocked, infection is blocked. In case of oral polio vaccine, it relies on the production of IgA production, and so you have a good mucosal immune system that is generated. Antibodies are efficient in preventing infection. Now you have also cell mediated protective mechanisms, as one of the ones that was discussed is a good robust CD8 positive response, because CD8 positive response will lyse the virally infected cells. Now in order to generate a good CD8 positive response, you also need to have CD4 help around, and so you need both of which would be required for this state. And on top of that, you have natural killer cells, because natural killer cells would kill the virally infected cells. And what is also shown over here, IL-2 is important, because interleukin 2, once T cells are activated, they will produce IL-2, and IL-2 is important for expansion of CTL precursors and so on. And what I am going to show you over here is the process of opsonization, and these are virus coated particles that are bound with antibodies, and you can see that these antibodies help opsonize, means they help the uptake of these viral coated particles into phagocytes. Okay, and then these phagocytes are taken up, and then they are sort of they are engulfed, and they are degraded in lysosomal compartments. Okay. Both CD4 and CD8 responses are important in case of viruses, and these are, and there is no doubt about it, because in case you have a compromised CD4 response, you will be unable to generate a good CD8 response, and if you do not have a good CD8 response, you are unable to kill these viruses. Okay, so it involves a combination of the two, and that is pretty much what is shown over here. You have the virally infected cells, and you have the specific T cell receptor, and this requires good recognition. That particular T cell has to recognize a virally infected cell expressing the MHC, and along with some viral encoded peptides, the TCR needs to be specific for it. So you are able to generate a good response, and then the perforins and the granzymes are secreted by the CD8 positive T cells, which would kill these virally infected cells. So what would also happen along with this is that you have activation of caspases, and the virally infected cells you would trigger apoptosis in these, and they would be degraded, and after degradation they would be broken up into apoptotic bodies, which would be phagocytosed. And we had discussed the efficient cleaning up of phagocytic cells in our class on T cell survival, and that is precisely what is shown over here. That you have good phagocytic cells which do a very efficient job of cleaning up apoptotic bodies. Okay. What is shown over here is the two paths that's important over here. You have the CD4 positive cell, which is recognizing which has a TCR that recognizes MHC class 2, and you have a CD8 positive cell that recognizes MHC class 1, and both of these are required. But what is important to note over here is that often in this case, in case of the class 1 response or the CD8 positive response, you have viral DNA, it is made into mRNA, and this mRNA is transcribed into viral proteins in the cytosol, and a part of these are then broken down and transported into the ER, and where they get loaded on MHC class 1, and then they are shown over here. So, you can see when we had discussed our class on MHC class 1, MHC class 2, this is why it is so important that you have the two parts over here. Over here, if the viral proteins are made in the cytosol, then perhaps some of them are degraded and the peptides are transported into the ER where they get loaded on MHC class 1. However, if some of them are digested in lysosomal compartments or MHC 2 like compartments, then chances are high that some of the viral peptides are loaded on MHC class 2 and will be showing those particular peptides to CD4 positive cells. Suffice to say, you need a good both CD4 as well as a CD8 response to generate good antiviral T cell responses. Okay. We will now discuss a particular virus which is extremely important and something that I am sure you are all familiar with. Okay, this is the human immunodeficiency virus known as HIV. Now, HIV is a retrovirus belonging to the lentivirus family, which is a slow grower. A retrovirus means it is made up of RNA and it needs to transcribe and it uses a reverse transcriptase to get into DNA and then again to form its particles. Now, the first reported case of AIDS was in 1981 by Gottlieb and Fahey, who realized that there were a lot of immunocompromised cases that were affecting young gay members in the Los Angeles community. Now, the cases that when they first came were usually found to be observed, or these infections were associated with people in advanced age, older people whose immune system was compromised. Now, what was surprising over here that these cases were showing up in younger people, and further investigations revealed that these younger people belong to a particular community, the gay community, and that is how really you have to give credit to the doctors and the scientists who were able to make this association. And the first report appeared in mortality and morbidity reports, which is some sort of a classic, and students must pay a little close attention as to the characteristics and to the acuteness of the physicians and the scientists who were able to pinpoint and try and figure this out, because this is the first time that this was being shown up. So, we went from a world without AIDS to AIDS all of a sudden, and you have to give credit to the doctors and the scientists to be able to who are able to distinguish this. So, what happens in case of AIDS is AIDS cause depletion of CD4 positive cells, which means you have less than 200 CD4 positive T cells per microliter of blood, and this, because you of the lowered CD4 number of cells, you have clinical complications. You have Kaposi sarcoma, it's a type of tumor that takes over. You have B cell lymphoma. So, you see your immunity is compromised. So, now your tumor cells as well as infections and susceptibility to infection increases. So, you have infections by opportunistic pathogens like cytomegalovirus, mycobacteria, pneumocystis carinii, etc. Okay, students should also learn to differentiate between HIV positive and AIDS. This is a very important distinction. When you are HIV positive, means you have been exposed to HIV and you have been tested to be HIV positive by either RT-PCR or ELISA. But it does not mean your AIDS. You go from HIV positive to AIDS or acquired immunodeficiency syndrome once your CD4 count drops. So, it is very important to understand that HIV, whether you are HIV positive or negative, depends on the detection by RT-PCR or ELISA, and once your CD4 count drops, and that can be determined by using specific antibodies and using flow cytometry, once your CD4 counts drop, that is when you start becoming susceptible to tumors and other infectious common infections that occur. Okay, very important for students to pay close attention to this. There are two main strains of HIV, HIV-1 and HIV-2. HIV-1 is a faster progressing one, it has higher mortality, whereas HIV-2 has low mortality, it is somewhat slower progressing. And in case in both these cases, the primary reservoirs are somewhat different. HIV-1, the primary reservoir is the African chimpanzee, whereas it is the sooty magpie in the HIV-2. in HIV-1 it's distantly related to the simian immunodeficiency virus, whereas HIV-2 is more closely related to the simian immunodeficiency virus. Okay. Now there are several proteins that are encoded by HIV. Remember, HIV is a virus, and several of the proteins are polyproteins. You have structural proteins, the matrix capsid nucleoprotein, you have envelope protein GP120, GP41, GP120 as you know is important in binding CD4, which is a receptor, the enzymes, the reverse transcriptase, protease and integrase. Remember, protease is a target of and protease is important because it is important in cleaving these polyproteins, and so protease is also a target, as a drug target. And you have different accessory proteins, like you have the TAT, REV, NEF, and so on, and we will be studying a little bit about these. What is shown over here is you have these different polyproteins encoded by HIV, and you can see that they are broken down over here. So you can see you have Pol, which is actually broken down into reverse transcriptase, integrase and the protease. Okay. So and again that is how the viral, in several viruses the proteins are transcribed as polyproteins and then they get broken down, and that is where the protease part is very important over here, because it is important in breaking down these proteins so that they can function properly. Now the targets of HIV, which we will be coming to, are again reverse transcriptase and the protease, which is very important. Okay, and I said we would be discussing a little bit about this. Now NEF protein enhances the pathogenicity of HIV. So if you have HIV lacking NEF, it leads to attenuated clinical phenotype with the reduced viral loads. Now VIF is important. VIF is important because it targets a cytosine deaminase known as SEM15. So if you have virus particles, so what it will target that. Now if you have HIV that lacks VIF or encodes a non-functional VIF, then what happens is that you have a high rate of mutations in this. So the cytosine deaminase is a host response to take care of such kind of particles, and that is why perhaps HIV has evolved in such a way that it encodes or it has been selected to for these molecules like VIF, which will target the cytosine deaminase, and as it is a endogenous activator of viral replications. Now you have, in terms of HIV, you have different types of viruses. You have the R type and you have the R5 type and you have the R4 type. Now the primary infections in HIV occur through the macrophages or the M-tropic, or the M-tropic stands for macrophage tropic. They are responsible for clinical infections and they infect dendritic cells and so on. So once you have infections from the M-tropic, and the co-receptor over here is the CCR5 receptor, and then subsequently as HIV evolves in the particular body, they turn from a R5 into a R4 or a M-tropic to a T-tropic, because then they start infecting T cells. So it is a very important transition, and again students should be should realize this. In fact that's why I said CCR5 plays an important role over here. So if you have individuals that lack CCR5 or a 32 base pair deletion, in fact a very high number, almost 10% of Caucasians, they have a deletion in CCR5, and that gives them significant protection against HIV, because what happens in this case is, even though they are exposed to HIV, HIV is not able to primarily infect them because of the lack of CCR5. So you see, apart from GP120 binding to CD4, you also need CCR5 as a co-receptor, and together this helps the entry of the virus. If you do not have it, then it is a problem. And what happens later on is, from a CCR5, from a R5 it becomes a R4, because now you have, it needs to bind another co-receptor, which is the CCR4 receptor or the stromal derived factor 1 or the fusin, which is important in syncytia forming HIV isolates, and this, as I said, occurs later in infection. Now apart from the chemokine receptors, you also have the MHC molecules, the HLA B27, B57, which play an important role. So for example, B27, B57, these are MHC class 1 molecules. They are associated with reduced AIDS. That means they are more protective, whereas rapid disease is associated with HLA B35. Okay, IL-10 is also important for AIDS protection. A polymorphism in the IL-10 promoter reduces IL-10 and results in increased HIV risk. So, if you produce high amounts of IL-10, chances are better for you in terms of getting or being less susceptible to AIDS. Now, studies have also shown that a particular molecule known as the tripartite motif or TRIM5A is an important molecule in encoding resistance. Now, how does this work? So, the reason is this class is about host mechanisms, and so that is why we are going to pick a few like SEM15 and then the TRIM5A as to how they help. Now, TRIM is known as tripartite motif because it has different motifs, and there are three main ones that are important. One is it encodes a ring motif. A ring stands for really interesting new genes, and these are primarily E3 ubiquitin ligases. E3 ubiquitin ligases are important players in the ubiquitin proteasome system. And so, this is, it shows it is a E3 ubiquitin ligase. It is important in the degradation of proteins using the cytosolic protein degradation route. It also has a B box which is involved in ubiquitination ligation specificity, and you have the coil domain which is involved in protein protein interaction. So, what happens in this case is that TRIM is rapidly autoubiquitinated and gets degraded by the proteasome. Now, once TRIM, what it also does, it binds to these viral capsids, and once upon binding of these viral capsids, it gets degraded, and so along with it the viral capsids are also degraded. So, it is a way by which the molecule is produced, it binds to viral capsids and gets degraded, and along with it degrades the viruses also. And in fact, this is done before the virus can reverse transcribe, and if you inhibit the proteasomal part, then of course TRIM cannot function because it is not degraded. So, that is an important aspect, and that is shown up over here. So, you have TRIM, TRIM5A, it is ubiquitinated and it gets degraded by the proteasome. Now, in case you are inhibiting the proteasome, then it is not degraded and you have these viral capsids being produced. Over here it is degraded, so the viral capsids are not being produced. Here the proteasome part is being blocked, and therefore you have viral capsids being produced. So, it is an important aspect on how the host has also evolved mechanisms to take care of viruses. Okay. Now, this is an interesting study that was done among a cohort of sex workers in Africa, now who were repeatedly infected with HIV for 15 years, but did not come down with disease. So, then scientists tried to find out as to what made them resistant to HIV. What was found is very interesting. What was found is that they had high IgA in mucosal surfaces. So, good IgA responses maybe thwarted HIV infection. They had high levels of IL-2 and HIV specific cytotoxic T lymphocytes were found. Now, also what was found is that their MHC phenotype was a little bit different. HLA-A6-802 showed twofold resistance, whereas HLA-B18 was able to resist infection. Okay. So, perhaps it was a combination of these factors, the MHC, the ability to generate CTLs, the ability to generate good mucosal immunity because of high titers of IgA together gave them immunity. What is interesting is that some of these workers developed AIDS after stopping their profession. So, why do you think so? I think students should think a little bit about it. What is happening is when they were continuing their profession, they were being exposed to their antigen, as a result of which their immune system was getting stimulated constantly. Now, what happened is once they stopped their profession, the exposure to antigen became less, and perhaps there was some down modulating of their immune system. As a result, some endogenous viruses that were kept inactive sort of became that were latent, in fact that they sort of re-emerged and they were able to maybe take over. So, it is very important that constant triggering with antigen may play an important role in keeping our immune system active. Now, it is generally accepted that reduced CD4 positive T cell susceptibility to infection and neutralizing antibodies do not correlate with resistance to HIV. So, and so therefore, it is important to understand that there are different factors that are sort of playing a role over here, and that is what is shown over here. You have the HLA type which plays an important role, you have cytokine gene polymorphisms, you have drug therapy. Meanwhile, because you are giving drug therapy, so you are selecting for certain types of viruses. You have chemokine receptor. I talked about the 32 the deletion in 32 base pairs in the CCR5, and then you have the NK receptor genotype. So there are different combinations that play an important role in finally determining the outcome of HIV, and it is very important to understand these different factors. So again, what is shown over here is that you have different players. You have the NK cells, you have the CD4 positive cells, the dendritic cells, all sort of coming together to play important roles. What is shown over here is the interaction between a particular MHC and a viral this is MHC class 1, and which is shown on which is being recognized by CD8 positive T cells, and if you have the appropriate combinations, maybe some viral peptides bind preferentially to certain MHC class 1s, whereas and may help in generating protective responses, whereas there are other MHC molecules that do not bind to some of these and are unable to generate efficient protective responses. Okay. And this is again shown over here in terms of the MHC class 1, and so what is shown over here is these particular ones, they reduce the risk of HIV, whereas some others are involved with Kaposi sarcoma risk, so on. So there is a combination of factors that are involved. Suffice to say that the HLA alleles or the MHC alleles are linked with immunoregulatory genes, and there are several variety of them which are important in determining susceptibility. Okay, some more that is shown over here. You have the host, this is the exposed population. Here is the CCR5 delta 32. You have rare HLAs over here, and so these are important in establishment of infection. So once you establish, then you have the MHC class 1 molecules that are important. Subsequently, you have the KERs, which is the killer inhibitory receptors, which is the NK receptors, which play an important role, and then you have the DRs or the MHC class 2, which are important in generation of different responses. So at different parts you have this modulation of the host responses along with the viruses, and when you have different aspects which contribute to susceptibility. And overall it would appear that if that CD4 without doubt CD4 positive T cell helps, CD8 and particular HLA alleles like B27 are important in determining susceptibility to HIV, and the ones that are maybe not that important are neutralizing antibodies, because it is very hard to generate neutralizing good neutralizing antibodies against HIV. You are soluble factors, innate restriction factors, and the ones then NK cells are probably potentially involved over here. But clearly the ones that seem to be important are here, the ones listed over here, because remember in case of HIV the primary response that is generated is the cellular response. So you have to focus on that. You have the CD4 positive response, the CD8 positive response, and the particular HLA alleles. Now, I am just briefly going to talk about the drugs against HIV, and the mechanisms are, as I have said, you have ones that mainly affect the RT or the reverse transcriptase, and you have nucleoside analogues, and which will affect the reverse transcriptase activity, and then you affect the protease, and I have mentioned the way by which it works, because you need to cleave HIV proteins into polyproteins. Now, the highly active antiretroviral treatment is a combination of these drugs of both the anti-RT and the anti-protease, which is required to reduce HIV numbers. Now, the fact is these drugs work. They work. They reduce HIV numbers. And remember, heart is expensive. It works on replicating viruses, but the reservoirs remain. But the drugs work. But they are highly expensive. Okay. And so, what about vaccines against AIDS? Now, the problem with vaccines is that the HIV mutates a lot. It is a very smart virus. It mutates, it has a very high mutation frequency. So, therefore, to generate a good antibody response, by the time an antibody response is generated, the epitope has changed. So, it becomes difficult to manage the new viruses. So that is one of the big problems. There is a high mutation in the envelope protein, it is difficult to get neutralizing antibodies. The other important parts is there is a lack of a good animal model, as all monkeys do not develop AIDS, and identification of immune strategies that lead to long lasting and efficient anti HIV CTL responses. These are all important aspects. Okay, so there are different strategies that have been listed to target AIDS or HIV, and different strategies have been followed, but there has always been some problem or the other. One hopes that better study will lead to mechanisms by which we understand this in a much better way. Okay, so we should be hopeful. Instead of being completely cynical, we should be hopeful that better work may lead to strategies by which we can control HIV, which is a very important disease. Okay, so we will now move on to some other aspects. So, apart from viruses, I will try and cover parasites and the newly emerging diseases. So, in case of parasites, the parasitic infections, now protozoa, helminth parasites, now these sort of enter the bloodstream and they are able to survive. Several of these target the gastrointestinal tract, and where they reside and they play important roles, worms for example. So, some of the features are that parasites in the human host are usually resistant to complement, okay, and macrophages can phagocytose protozoa, but somehow they are resistant to these to macrophages, complement, so on. So, that makes it a little bit difficult. Now, cell mediated immunity is the primary defense against protozoa that survive within macrophages. Okay, so, they replicate and you are able to generate a specific CTL response. Now, IgE antibodies and eosinophils mediate defense against many helminthic infections. So, again IgE responses are very important. Remember, to get a good IgE response, you would need a good Th2 response. So, that is again important. Okay. Now, one should also be a little bit careful that when you are generating these responses, it can lead to tissue injury, because macrophages can get activated and they can induce a delayed type hypersensitivity response, and then may result in the formation of granulomas. And these granulomas, you could see the tubercles are actually tubercles during infection with tuberculosis is actually they are granulomas, and they are a host response, and it causes damage, host damage. Now, cytokines are important. Now, in like TNF alpha is very important against innate immunity to T brucei, and now what happens is TNF alpha binds and is internalized by T brucei. Now, what is interesting is only parasites isolated during the peak of parasitemia are lysed by TNF alpha, but the mechanisms are unclear over here. Now, this one is to show you the different mechanisms by which that are involved. Okay. So, this is to a parasite infection, and you can primarily see that what you have is expulsion, and to get expulsion you need increasing secretion of the mucus. So, this is what I meant, this you need expulsion over here. Now, now for increased secretion of the mucus, your cytokine profile has to be primarily Th2. So, which is shown over here, you are generating a specific T cell response, you have non-specific inflammatory stimuli which secretes the goblet cells, the increased secretion of mucus, you have these the IL-4, IL-10, IL-5, these are all Th2 producers. These produce antibodies, and these nematodes are produced. The mast cells stimulate IgE production, and what you are doing is trying to stimulate these and expel them. So, now what would happen if you had a Th1 response, for example? Now, what a Th1 response would be pro-inflammatory. What it would do is it would actually oppose the secretion of the mucus and so on. So, what would happen is they would remain stuck, and you would generate an inflammatory reaction. and you would have granuloma formation over there where things would be stuck, it would cause enormous damage and hurt to the host. So, a Th2 response in this case, what it does is it helps expulsion of eggs, it helps expansion of nematodes, so that there is increased secretion and these are expelled off. But of course, it is conducive for the nematodes to be there and replicate and try and spread their progeny because you have this Th2 response. So, it is very important to understand, and it also signifies how the Th1, Th2 are important in playing an important role in this process. Okay, and that is what is shown over here. You have you have different cytokines being involved, but primarily it is the Th2 response which through the generation of IgE they help in secretion of the mucus by the goblet cells, and as a result of which it helps the expulsion. What it also is important is the innate cells act as the first line of defense, and later on the T cells come in for specificity and they help in this particular process. Okay. Now, we will talk a little bit about emerging infectious diseases. Why is this important? Okay. Now, if you have read the newspapers in recent days, you have these newer diseases that are coming up. You have the SARS, and then you have the H1N1, you have H5N5. Why are these coming? I think students should think a little bit about this. Why is it that you have every 2-3 years a new disease that is emerging or a new virus that is emerging? One of the thoughts is that as the human population is expanding, we are venturing into areas and trying to take over areas that where viruses have lived in some sort of a equilibrium with their environment. Now, as our numbers increase and we try and disturb that, they are also making changes and in response to the changes in their environment, and what they are trying to do is to expand their repertoire and trying to jump. So, in case some of these are successful, what they will do is they are successful in jumping and infecting humans. So, what happens as a result of which you have newer diseases. So, this is some aspect that students should be thinking about, because as we venture into newer environments, as our population increases, we are moving into newer territories, and this is exactly what happens. If you remember, in the last class I gave you an excellent example of what happened to the endogenous American Indian population, who were never exposed to Mycobacterium tuberculosis or Mycobacterium leprosy before. When the Europeans invaded or entered America for the first time, they came along, they also brought with them diseases. So, a lot of the endogenous American Indians had very poor resistance to Mycobacterium tuberculosis and mycobacterial diseases and several of them were actually they got this disease and they were highly susceptible to these diseases. You had several people dying because of their inability to generate a proper host response, because they were not being exposed to it, whereas we have been sort of living along with Mycobacterium tuberculosis. Our immune system is exposed to it, and so we sort of have a heightened immune response, whereas these people were not exposed to it, and they were highly susceptible to these pathogens. It's something that to remember. So, what is happening now is pretty much the same. The different influenza viruses are sort of mutating and changing over their host preferences from pigs, or from birds to humans, as a result of the changes that are occurring in their environment. And also, given the fact that these days people travel a lot, and so you can move into different countries very quickly. So, they are you carry the disease along with you. And this is again an important aspect that we should all be careful about. Now, emerging infectious diseases are important, and in fact, you have the Center for Disease in Atlanta, which sort of monitors this, and one of the areas that they have been actively monitoring is a virus that causes, or a group of viruses that causes viral hemorrhagic fever, or VHF. and it over here it produces systemic and often fatal diseases. Now, the filoviruses Ebola, Marburg, Lassa fever, these belong to this category, and I thought we would end it with a little bit look into the future about emerging infectious diseases, because it has potential for students to sort of think about something that might happen in the future also. There have been books written on Ebola, and I think there is a book known as the Hot Zone, which again students should read. It is about what happens in a place where a chimpanzee center near Washington DC, that is actually in the state of Virginia, where you have an outbreak. And how the CDC combined with other scientists try and contain that outbreak, because if it goes out of control, then the disease will spread and will cause fear and panic, which you don't want. But it's a very it's a very interesting book, and it's something that students should also look and think about, because apart from coursework, you should be thinking about these other different possibilities that may be occurring. Okay, so a little bit about viral hemorrhagic fever. So, they are a different group of viruses, and what they do is they cause systemic and frequently fatal disease. Now, the best virus that is known is the Ebola. Now, but there are different other members of it. You have Marburg, Lassa fever, yellow fever, Rift Valley fever virus, so on. So, what is seen over here is you have the virus, and it causes increased endothelial porosity edema and lymphocyte apoptosis very quickly, and it's a systemic infection that takes place, and that what it does is basically what is happening is you have decreased production of clotting factors, as a result of which you have hypotension, because your blood pressure drops rapidly. And the example is Ebola, which we will discuss, and it causes lethal hemorrhagic disease, flu-like symptoms, rapid fever, bleeding, shock, death. But now, what is surprising over here is about 70% of the patients die, whereas 30% survive. So, a question that can be asked is, how come 30% of the patients survive? How are the survivors different from the ones that are susceptible? Okay, it is very important for us as immunologists, as people who are interested in the host response, to be asking what differentiates this group, this cohort from the other. And it's very, and because if we understand this, then it might shed some light. In fact, this is precisely the type of studies that were done with HIV, where you found that a person who has been repeatedly in contact with HIV positive people was not coming down with the disease, and they're not coming down with the disease because of the mutation in CCR5. In fact, that's how the 32 base pair deletion was found out. And because it can shed important light as to what makes, or what are the attributes of survivors, or what are the attributes of a person who is more susceptible. So, if you understand these, maybe there could be ways by which you could make the person who is more susceptible into and reduce the susceptibility, so and increase the chances of survival. So, it's very important in terms of research, and it has translational applications. Okay, so this is a structure of the Ebola virus. I think the whole idea is that it causes rapid cytotoxicity, as mentioned, a very high fever. There is a lot of bleeding, and a study on susceptibility was actually done. In fact, in a study that was published in Nature Medicine several years back, what was found is that the non-survivors were unable to generate a proper humoral response to the virus, a very rapid and fast humoral response. Also the non-survivors not able to maintain cytokine levels, that is, they had an impaired T cell response, and the lack of a proper B and T cell response led to increased apoptosis of peripheral blood mononuclear cells and death of immune and non-immune cells in non-survivors. What also happened is you also had rapid release of vasoactive agents, including cytokines, chemokines, histamines, and as a result you had what is known as a shock-like syndrome. We had discussed this in our studies on septic shock during the innate immunity class. So, it's very important. So, if you so it's very important for students to be able to understand this. Okay. So Ebola was was important in our study because it is an emerging virus, and the last part of it was to tell you about the cohort of survivors, the differences between the survivors and the non-survivors, and it tells us these sort of studies may lead to better ways by which you can increase the survivability of the group that are non-survivors, because once you understand what are the factors that make it, perhaps we can think of better ways, or maybe use adjuvants, vaccines, by which you can boost up the immunity. Okay. So, overall this class sort of dealt with the host response. In fact, the previous as well as this is very important, because actually the entire immune system is coming together. You can see that CD4 cells are important, CD8 cells are important, NK cells are important, B cells are terribly important because they are very important in generating neutralizing antibody responses, and overall you have these main players. But it is also important to understand some of the key ones that we discussed. We discussed the JAK-STAT pathway, which is very important in terms of responses to interferons, very it's and certain chemokine receptors that are important in generating our ability to be resistant to certain viruses, the CCR5. So, these are important aspects because they are important players in trying to understand how we resist resist different pathogens that we are surrounded with. Okay. So, I will just briefly summarize this class. So, we first started off with viruses, and we discussed the fact that they must replicate to try and spread, and there are different ways by which you can target. You can target the receptor, you can target the progeny, you can target the reverse transcriptase, you can target the protease, so on so forth. There are different ways by which you can do that. We also talked about the interferons, the neutralizing ability to generate interferons, the JAK-STAT pathway, and the way type 1 interferons work versus the type 2 interferons. The type 1s are primarily antiviral. The type 2 interferon gamma is against intercellular infections. If you do not have interferon gamma or its receptor, you are highly susceptible to BCG infections, that was discussed. Now, in terms of viral or in terms of vaccines, the ability to generate neutralizing antibodies is really the key, and this cannot be overemphasized. All the successful vaccines are the ones where you generate a very good neutralizing antibody. We also looked at the role of T cells. You need both CD4 and CD8 to play an important role in generating this response. We studied HIV in great detail, and I want students to be very aware of the differences between HIV positive and AIDS positive. You turn from a HIV positive to AIDS positive only once your CD4 count goes down. Okay. And so the assays to determine these two are different. For HIV positive, you can do it by RT-PCR or ELISA. For AIDS, you would have to find the numbers of your CD4 positive cells, which is why you need to understand the importance of cell surface antibodies and the use of flow cytometry over here. Now, what this does is the lowered CD4 count makes you highly susceptible to tumors, to endogenous tumors, and susceptibility to intercellular pathogens. And in fact, some common opportunistic pathogens that we live with, once our immunity goes down, they sort of take over. Now, in the host factors that is important, we discussed the importance or role of MHC. You could see that having certain MHCs was protective, whereas others were more susceptible. We studied the importance of chemokine receptors, the CCR5 to CCR4 transition. The infection of HIV starts off with CCR5, the M-tropic, and then the virus changes over to CCR4 or the T-tropic viruses. We studied the importance of the TRIM5A proteins, which is important because they sort of they bind to the viral capsids and target it for proteasomal destruction. We also studied how the virus encodes a factor known as VIF, which binds to SEM15, which is a cytosine deaminase, which is important. So, these are important. You can see this evolving nature of pathogens and the host proteins, which play an important role. We also studied the role of parasitic infections, especially nematodes, with the important role of Th2 type and secretion of mucus, so that you can extrude these organisms. Then finally, we studied emerging diseases with Ebola being an important component, and the difference between survivors and non-survivors. So, overall I hope this has been very useful to you, and I hope you will be, whenever you read newspaper articles on disease outbreaks and all, you will be thinking about this class and you will be able to link it together. If you are able to do that, then that will mean that this class has been successful. Thank you so much.